Genetics of behavior and natural selection. Just as a thorough understanding of an animal's morphology requires knowledge of how it develops before it hatches from an egg or emerges from its mother's womb, a complete understanding of an animal's behavior requires knowledge of animal's development during its lifetime. To gain this knowledge, one asks how the individual's genes and its experiences causes it to behave as it does. The ontogeny of behavior is a subject which arouses considerable interest, perhaps because of the seeming contrast between humans and other animals, in how behavioral skills are acquired. Whereas humans extensively adjust their behavior based on experience, that is through the process of learning and learning set learning, the behavior of many animal species seem to be automatic, that is innate or stereotyped, as if it were pre-programmed. Introduction. There have been intense clashes of opinion in behavioral developmental studies. Prior to the 1960s, there existed a profound disagreement between European, particularly German ethologists and American psychologists regarding methods and interpretations of such studies. The ethologists, with the help of many examples of animals, described complex behavioral patterns in response to particular stimuli under circumstances that seemed to exclude the opportunity for learning. Indeed, learning which is based on external influences was opposed with genetic control of behavior which was based on internal influences. Austrian zoologist Konrad Lorenz, who won a Nobel Prize for his ethological studies, went so far as to classify behavioral patterns into two distinct categories: acquired, that is learned, and innate, means stereotyped. An example regarding innate behavior is adult herring gulls, which have a red spot on the lower tip of the bill, and when these birds have food for their chicks, the adult point their bill downward while waving it slowly back and forth in front of the young. Newly hatched chicks, which accurately peck at the red spot on the parent's bill, suggesting that a herring gull chick possesses innate, that is genetically based knowledge of where to peck for food. Ethologists termed pecking behavior as a fixed action pattern to indicate that it was performed automatically and correctly for the first time. It was elicited, apparently regardless of the animal's experience. The psychologists, in contrast, assumed that the experiences with the environment, that is learning process, were the main or even exclusive determinants of ontogeny. Accordingly, they saw nothing in the pecking behavior of herring gull chicks that could not be explained by learning while still in the egg. Conditioning or by trial and error learning, for example, chicks might learn to peck before hatching as a result of the rhythmic beating of their heart, or they might have a pecking reflex and simply learn to associate a food reward with pecking at the parent's bill. Moreover, a chick's pecking accuracy improves with the age, and after about 2 days it requires, in addition to the red spot, the complete configuration of an adult's head and bill to elicit pecking. What the acquired innate dichotomy observed is that learning is possibly only after the animal has already been steered by its genes to develop its behavior in a certain way. An animal may well learn, but which experiences are important to the development of its behavior depend on those that have promoted the genetic success of its ancestors. Equally, whatever experiences an individual already has or had can influence how its genes are activated and thus can affect their subsequent role in shaping its behavior. Modern animal behaviorists see the stark dichotomy of acquired versus innate as far too simplistic. No behavior is either strictly innate or entirely learnt. Rather, all behaviors are a result of complex interaction between genes and the environment. Biologists have always been fascinated with the question of where the traits that exist today came from, that is, their evolutionary history. However, the exploration of the history of behaviors and their underlying mechanism is exceptionally challenging. Unfortunately, the fossil record is largely uninformative. Only under rare circumstances, such as the discovery of a fossilized dinosaur nest topped by an adult, a situation suggestive of a parental care, is there sufficient information captured in fossils to enable paleontologists to draw inferences about the origin and subsequent evolution of complex social and reproductive behaviors. As a result, it has been necessary to develop alternative and indirect approaches to infer evolutionary histories of behavior. Character mapping. The first approach, called character mapping, begins by constructing a phylogenetic tree, that is, a depiction of the presumed relationship of a species of interest to its closest living relatives. Phylogeny refers to the evolutionary history of one or a group of interrelated species. Hypotheses regarding phylogenetic relationship often are based on similarities among existing species in morphological traits and DNA sequences. Once the phylogenetic tree is established, character traits or behaviors such as parental care of the extant species are attached or mapped to it. Sites on the tree called ancestral nodes are drawn where changes in the behavior of interest apparently occurs. This is accompanied by minimizing the number of character state transitions or changes necessary to account for all the diversity seen among related species today. In other words, the shortest evolutionary path taken by any character from its origin to the present is considered to be the most parsimonious or thrifty and therefore the most likely. Assuming that the behaviors of extant species have remained the same since the last speciation event in their lineage and that the shortest evolutionary path is indeed most likely, a hypothesis can be formulated about the relative timing of origin of various behaviors and their subsequent loss or evolutionary modifications. These assumptions are most valid for complex behaviors whose evolution requires many improbable changes rather than highly variable, that is, plastic behaviors. Moreover, it is more reasonable to suppose that a complex behavior, one that is shared by two or more species, was present in a common ancestor and later on it evolved multiple times independently. Phylogenetic reconstructions and character mapping have been used to infer the historical trajectories of the male secondary sexual characters and female mating preferences in several taxa such as Central American frogs and swordtail fishes. In the frogs, electrophysiological studies of present-day species indicate that females have identical auditory preferences regardless of the acoustic characteristics of the mating calls of male. The most frugal hypothesis, therefore, the question is that whether female preferences evolved first, that is, they are ancestral or older, and that the male calls evolved secondarily in some species to take advantage of these pre-existing preferences. In the swordtail fishes, females in the species with and without swords prefer males with artificial swords attached to their caudal fins over unsworded males. The hypothesis that ancestral females possessed the preference for a sword-like structure is more prudent than that the preference for swords evolved multiple times independently in the lineage of each existing species. Drawbacks of character mapping. One general problem with the character mapping approach is that the most parsimonious evolutionary pathway may not be most likely. Evolution itself is a slow progressive change over a time, and evolutionary change is seldom unidirectional. So, small changes in the characteristic in one direction or the other may have occurred multiple times over the evolutionary history of a species group. A more specific problem, while inferring with the evolutionary history of sexually selected characters using character mapping, is that it is often difficult to determine exactly what aspects of a male trait the female prefers. Behavioral genetics. The evidence is now compelling that genes influence behavior in all animals, including humans. Indeed, an increasing share of biomedical research is devoted to the hunt for genes involved in human behavioral maladies such as alcoholism, obesity, schizophrenia, and Alzheimer's disease. These studies are often pursued using animal models with subjects like mice, rats, and dogs with behavioral symptoms resembling those of humans. It is therefore unfortunate that the idea that genes affect behavior is the subject of much heated and confused discussion. The principal point of confusion arises from equating genetic influence on behavior with genetic determination of behavior. To do so is to mistakenly believe that identifying genes for behavior implies that the gene controls fully and inevitably this behavior. In actuality, to say that there are genes for a particular behavior means only that when a population of individuals there exists genetic variation underlying some of the differences in this specific behavior. To cite an example involving a morphological trait, the statement that there are genes for coat color in guinea pigs or horses means that genetic variation in guinea pig or horse population is responsible for some of the variation in coat color. Furthermore, identifying a gene that influences a behavior does not imply that the behavior is inevitable. There is considerable variation among behaviors and the relative importance of individuals' genetic constitution and its environment to the expression of the behavior. Occasionally, the possession of a particular form of a gene does not consistently result in the individual having a particular form of a behavior. More frequently, however, form of the behavior is due to a complex interaction between genes and environment. The strength of the influence of a gene on particular behavior is quantified by a genetic measure called heritability. Heritability is defined as the fraction of the total variation in a trait among individuals in a population that is attributable to the genetic variation among those individuals. The remaining source of variation is to, of course, the environment. Values of heritability range between 0 and 1, and the smaller the environmental variation experienced by the individuals in a population, the greater will be the fraction of the total variation in the behavior that is the result of genetic variation. The degree of genetic influence on a particular behavior is not a fixed characteristic. Rather, heritability can vary greatly depending on how much environmental variation is experienced by the individuals in the specific population being studied. Thus, regarding the calling behavior of male crickets, if every male fed well, thereby eliminating several environmental influences on calling, the numerical value of heritability would be considerably higher. Numerous studies involving diverse species, including humans, have detected some level of heritability for every trait that has ever been examined. For example, the mean value of heritability for morphological traits such as body and wing length is 0.46. For life history traits such as fecundity and lifespan, it is 0.26, and for behavioral traits such as calling duration and fighting stamina, it is 0.3. Thus, the genetic influence on the characteristic of an individual animal falls generally between 30 and 50% for most of the traits. Natural selection in action. The most straightforward way to study the function of a behavior is to see how natural selection operates on it under current conditions by studying differential reproduction. Often this kind of investigation can be conducted by exploiting the naturally occurring variation among individuals, such as in a particular phenotypic observable trait in a population. Sometimes, however, the researcher must experimentally enhance behavioral variation where too little exists in nature. The experimental approach may have the disadvantage of involving unnatural variants, but it has the advantage of revealing how differences among individuals, even in a single trait, can cause variation in reproductive fitness. Either way, a study of natural selection acting on behavior requires that the researcher be able to observe natural populations and obtain detailed information on each individual's survival, its ability to attract a mate, its fertility, and so forth. All of this information is essential to assess an animal's success in passing on its genes. An investigation of why male titmice or great tits, woodland birds of Europe, sing multiple songs serves to illustrate how a behavioral function can be studied by exploiting naturally existing variation. Each great tit male has a repertory of 1 to 8 songs that he uses to advertise his presence on a territory. Investigators can acquire detailed information on the breeding biology of these birds. Because great tits are cavity nesters, that is, they readily accept man-made nest boxes. In one experiment on a wooded estate near Oxford, England, English zoologist John Krebs and his colleagues installed and regularly inspected nest boxes during the breeding season. The researchers recorded the singing behavior of each breeding male in order to determine repertory size. They also recorded the egg-laying date, the clutch size, that is the number of eggs, the brood size, number of youngs, and the fledgling weight for the nests of numerous males. It was possible to monitor the survival of each male's young to the time of its own breeding because all the young were banded before they fledged, and most fledglings returned to the same woods to breed themselves. The researchers found that the individual teats had different repertory sizes. Male with larger repertories had chicks that were heavier at fledgling, and more of these chicks survived to breed than offsprings of males with smaller repertories. Thus, male repertory size and reproductive success were correlated. The underlying mechanism is that males with larger song repertories were able to acquire superior territories, specifically ones with better food. Previous studies had shown that size and survival of the young depends on body weight at the time of fledging. The bigger and heavier the fledgling, the greater its chances of survival to maturity. Thus, the function of a great tit male singing multiple song is to help him secure a top quality breeding territory and mate. So why do all males not sing multiple songs? Perhaps songs are learned over time, so that only the oldest males can possess a large repertory. Alternatively, perhaps there are costs such as time away from foraging or increased vulnerability to predations to singing multiple songs, and only the biggest, strongest males can sing many songs and still survive. Direct comparisons of the individuals of the same species exhibiting natural variation in behavior is a revealing way to study behavior function. However, when appropriate natural variations do not exist, experimental manipulations can provide the needed variation in the behavior. The variant forms are then studied in the field to determine how well extreme forms of behavior do in the face of natural selection. Using this method, American biologist Thomas Seeley investigated nest site choice in a species of Southeast Asian honeybee. Colonies built their nests of beeswax comb amid dense foliage suspended from the branches of bushes and understory trees. Moreover, if a colony's nest loses its cover during the dry season, when many trees shed their leaves, the colony will build its new nest in another leafy site. What is the function of this behavior of nesting in dense vegetation? Is it to prevent the nest from overheating under the strong tropical sun, or to conceal the nest from predators, or both? To test the anti-predator hypothesis, pairs of naturally occurring colonies were identified. Within each pair, the vegetation around the nest of one colony, which served as the experimental unit, was removed, leaving only enough to provide shade, but rendering it conspicuous to predators. The vegetation surrounding the nest of the second colony, which served as the control, was not removed. Measurement of nest site temperatures one day later revealed no significant differences between the two nests. Within 1 week, however, 4 of the 7 experimental colonies had been discovered and destroyed by the predators, probably monkeys and tree shrews, whereas none of the controlled nest had suffered any damage. Thus, it appears that Apis florea colonies choose dense vegetation as nesting sites primarily to conceal their nests from predators. Another example of a well-controlled field experiment on the function of behavior is Dutch-born British zoologist and ethologist Nicolaas Tinbergen, pioneering studies of eggshell removal by black-headed gulls. In a matter of hours after their eggs hatch, they pick up the empty eggshells, fly off and drop them well away from the nest. Why should a gull engage in this behavior? One hypothesis was that the sharp edges of the shells might injure the chicks, a danger that is well known to poultry breeders. Another hypothesis was that the white inside of the broken shells might attract the predators such as crows and herring gulls flying overhead and so endanger the brood. To test the latter hypothesis, Tinbergen and his colleagues distributed single gull decoy eggs around dunes where the black-headed gulls nest and placed broken eggshell near some of the decoy eggs while leaving others isolated. The investigations found that the eggs near the broken shells were preyed upon sooner than the isolated, less conspicuous eggs. Evidently, the removal of broken eggshells from the nest by the gull helps to maintain the camouflage of the brood, thereby reducing predation.